Hello students, this is going to be the review for exam 2. I'm sorry about this getting to you a little late, and I know that exam 2 is today, but so you can watch this before you take the exam. But anyways, so let's just go over a few things before you get to the exam. So in chapter 6 we talk about energy, exergonic and endergonic reactions. So endergonic reactions are not spontaneous. Energy is absorbed, and free energy is more than zero, right? If free energy is more than zero, it's going to be not spontaneous, right? So endergonic, meaning that it's absorbing energy. Exergonic reactions, meaning the energy is released, right? Exergonic, you can think of it as heating the environment. It's going to be heating the area around it. So reaction is spontaneous. That means that free energy is less than zero, right? So free energy is less than zero. It means that it's going to be spontaneous. Any other type of energy stuff, so energy coupling, ATP is hydrolyzed, right? So this is phosphorylation. So any type of phosphate group is added to a molecule. That's called phosphorylation. This is happening when energy coupling is occurring. So you have ATP coupling onto a molecule. That's called phosphorylation, and then something else is happening at the same time. Enzymes help a reaction occur, but they're not changed by the reaction. So they don't alter the free energy of the reaction. They're not changed by the reaction so that they can be reused. So enzymes can be reused over and over again, but they do change the activation energy. They don't change the free energy, they alter the activation energy. There is something called enzyme specificity, right? So a substrate has to bind to an active site. That active site has a specific place for a substrate. If another substrate comes in, it's not going to bind to that specific active site. Catabolism is the process of breaking down complex molecules into simpler compounds. And anabolism is the process of building complicated molecules into simpler ones. So building is anabolism, catabolism is breaking apart. Oxidation is the process of losing electrons, and reduction is the process of gaining electrons. Right? Oxidation is losing, reduction is gaining electrons. To break a chemical bond between atoms, energy must be added. So to break this bond, you have to add energy to it. That's enough with energy. Let's go into cellular respiration. Mitochondria and ATP. So you should know the processes of cellular respiration. So we just talked about oxidation and reduction. So oxidation is losing electrons. Reduction is gaining electrons. You should think about that oil rig. Oxidation is losing, reduction is gaining. Oil rig. All right. Glycolysis happens in the cytosol, or the cytoplasm. Okay? The breakdown of pyruvic acid. So the breakdown of pyruvic acid, or pyruvate oxidation, happens where? In the matrix. And then the next step, citric acid cycle, or the Krebs cycle, where does that occur? That also happens in the matrix. And then the electron transport chain is next. And then where does that occur? That happens in the inner membrane, or in the cristae, is what they call it. You should know the net gain ATP for those. The source of energy that drives ATP synthase is the proton motive force. Okay, or that chemiosmosis. Basically, hydrogen ions, right? Those protons. During glycolysis, glucose is broken down into what? During glycolysis, it's going to be broken down into pyruvate. Right? So all those steps, it goes down into pyruvate. If there's no oxygen, pyruvate can be converted into what? Pyruvate can be converted into lactic acid. Right? It won't go into the citric acid cycle chain. It'll be converted into lactic acid. All right, does mitochondria and chloroplasts have double membranes? Yes, they both do. That's enough of that. Let's go into photosynthesis. So photosynthesis is light energy converted into chemical energy. So first, do light reactions of photosynthesis require the presence of oxygen? No. So the light reactions don't require oxygen. They don't even require CO2. Light reactions don't require CO2. During photosynthesis, the light reactions occur in the thylakoid membrane of the granite. True or false? This is true. So the thylakoid membrane contains the chloroplasts. What is the wavelength of light that chlorophyll A molecule in this photosystem absorbs? So in photosystem 1, what wavelength of light does it absorb? Photosystem 1 absorbs 700 nanometers, and then photosystem 2 absorbs 680 nanometers. What are the main products of photosystem 1 when it's associated? The main product of photosystem 1 is NADPH. Okay, that's the main product of photosystem 1. So at the end, NADPH goes to the Calvin cycle. The main product of photosystem 2 is O2 and H. O2 is a byproduct, but hydrogen is the main product that goes to the ATP synthase. Where does energy come from that replaces the missing electron in chlorophyll A molecule of photosystem 1 and 2? So photosystem 1 the energy comes from light. And then photosystem 2, the energy can come from light, or it can come from the splitting of water, which comes from light. I think that's it for photosynthesis. Let's go to mitosis. We talked about ploidy with mitosis. So ploidy, we have haploid. Haploid means that it has one set of DNA, and then diploid has two sets of DNA. In what phase of mitosis do the homologous chromosomes split? That's anaphase, right? There is a picture on here that you should be able to know what the phase of mitosis is. So make sure that you know what you're looking at when you see the pictures. In telophase, the division of cytoplasm occurs. What is the division of cytoplasm called? That's called cytokinesis. All right. Y'all should know most of the rest of that. Meiosis. This is basically talking about crossing over events. So what phase does crossing over take place? That's in prophase 1. Also, you should know what the different phases do in meiosis. All right, make sure that you can figure out how the different phases are working, what the tetrads are doing in each phase. Okay, how they're separated. All right. If you have any questions before the test, please let me know. The test will be available until tomorrow at midnight. Okay, so get studying and get to it. And yeah, just email me if you have any questions. All right.